Hello, this is part 2 of chapter 27. In this section, which is concept 3, I'm going to talk about nutritional and metabolic diversity in prokaryotes. The prokaryotes exhibit some unique modes of nutrition as well as every type of nutrition found in eukaryotes. In addition, metabolic diversity is greater among prokaryotes than eukaryotes. Prokaryotes exhibit a great diversity in how they obtain necessary resources, energy and carbon, to synthesize organic compounds. Some obtain energy from light, and we call these phototrophs, while others use chemicals taken from the environment, those called chemotrophs. Many can utilize CO2 or related compound as a source of carbon. We refer to these groups as autotrophs, and others require at least one organic nutrient as a carbon source. We call these organisms heterotrophs. Depending upon the energy source and the carbon source, prokaryotes have 4 possible nutritional modes. Photoautotrophs use light energy to synthesize organic compounds from CO2. This mode includes the cyanobacteria. Actually, all photosynthetic eukaryotes fit in this category. The other mode of nutrition are referred to as chemoautotrophs. Those organisms require only carbon dioxide as a carbon source and obtain energy by oxidizing inorganic compounds such as hydrogen sulfide, ammonia, ferrous, and so on. This mode of nutrition is unique to certain prokaryotes, like archaea of the genus Sulfolobus. The third mode of nutrition, photoheterotrophs, use light to generate ATP from an organic carbon source. This mode of nutrition is unique to certain prokaryotes, such as especially those the aquatic halophiles. The fourth mode, chemoheterotrophs, must obtain organic molecules for energy and as a source of carbon also, found in many bacteria as well as most eukaryotes. Examples in bacteria include Clostridium, some protists, fungi, and so on, and animals, of course. The table shown summarizes the fourth mode of nutrition. You can refer to this table. The role of oxygen in metabolism. Prokaryotic metabolism varies with respect to oxygen. Organisms are classified into obligate aerobes, obligate anaerobes, and facultative anaerobes. Obligate or strict aerobes require oxygen for cellular respiration. Obligate anaerobes are poisoned by oxygen and live by fermentation or use substances other than oxygen for anaerobic respiration. That's to say, in their electron transport chain, the final electron acceptor is not oxygen. Could be sulfur, iron, etc. The third group, the facultative anaerobes, these organisms can use oxygen if it is present, or carry out fermentation or anaerobic respiration if it is not present. Nitrogen metabolism. While eukaryotes can only use some forms of nitrogen to produce proteins and nucleic acids, prokaryotes can metabolize most nitrogen compounds. Prokaryotes are extremely important to the recycling of nitrogen through ecosystems. Some chemoautotrophs, bacteria, like some of these bacteria, convert ammonia to nitric dioxide. Other bacteria, such as Pseudomonas, denitrify nitrogen dioxide or nitrate to atmospheric nitrogen. Nitrogen fixation, nitrogen transformation into ammonia, is unique to certain prokaryotes, cyanobacteria, and is the only mechanism that makes atmospheric nitrogen available to organisms for incorporation into organic compounds. The nitrogen-fixing cyanobacteria are very self-sufficient. They need only light, energy, carbon dioxide, nitrogen, water, and few minerals to grow. The roles of soil bacteria in the nitrogen nutrition of plants are shown in this figure. You can see we have different types of bacteria. For example, nitrogen-fixing bacteria fix atmospheric nitrogen into ammonia, the form ammonia. Then ammonia in the soil transform into ammonium. Another type of bacteria, nitrifying bacteria, transform ammonium into nitrate. And nitrate can be used by the plants. Also, we have ammonifying bacteria, which can obtain nitrogen from amino acids or other organic compound and transform it into ammonium. On the other hand, we have denitrifying bacteria that antagonizes the function of the others by transferring nitrate into atmospheric nitrogen, making nitrate unavailable or not available for the plants. Another rule of bacteria is shown in this figure, root nodules on a legume. The spherical structures along this soybean root system are nodules containing rhizobium bacteria, type of bacteria. The bacteria fix nitrogen and obtain photosynthetic products supplied by the plants. It's a symbiotic relationship. Metabolic cooperation. Cooperation between prokaryotes allows them to use environmental resources they could not use as individual cells. In the cyanobacterium, Anabenna or nostoc or any cyanobacterium, most cyanobacteria, photosynthetic cells and nitrogen-fixing cells called heterocytes or heterocysts exchange metabolic products. We can see this next figure, please. This figure shows filamentous cyanobacterium Anabenna. Notice the heterocytes or heterocysts fix nitrogen, while other cells carry out photosynthesis. Note that each heterocyte is surrounded by a thickened cell wall that restricts or prevents entry of oxygen produced by neighboring photosynthetic cells. Intercellular connections between heterocysts and photosynthetic cells allow heterocysts to transport fixed nitrogen to neighboring cells and to receive carbohydrates from photosynthetic cells. Metabolic cooperation occurs between different prokaryotic species in surface coating colonies called biofilms, and we talk about biofilms in chapter 9 in signaling, chapter dealing with signaling. Sulfate-consuming bacteria and methane-consuming archaea is another example. Those archaea are found on the ocean's floor, use each other waste products of metabolism. Now, we're going to talk about the fourth concept of this chapter. Prokaryotes have radiated into a diverse set of lineages. The origin of prokaryotes dates back to 3.5 billion years ago. Prokaryotes now inhabit every environment known to support life, as we said in the introduction. Advances in genomics are beginning to reveal the extent of prokaryotic diversity. Genetic analysis led to the division of prokaryotes into two domains, bacteria and archaea. A few taxonomic groups are monophyletic. It has a single origin, one ancestor, but others are scattered throughout several lineages. Advances in molecular techniques help to add new branches to the tree of life each year. This figure shows the three domains of life: domain bacteria, domain archaea, and domain eukarya. The use of the polymerase chain reaction PCR has allowed for more rapid sequencing of prokaryote genomes. A handful of soil may contain 10,000 prokaryotic species. Horizontal gene transfer between prokaryotes obscure the root of the tree of life. Bacteria include the vast majority of prokaryotic species familiar to most people. Diverse nutritional types are represented among bacteria, and bacteria have several groups, major groups, depicted on figure 2716. This figure is not required. Domain archaea. Archaea have many unique characteristics, and archaea share certain traits with bacteria and other traits with eukarya. The following table show this compare between the three major domains of life. As you know, nuclear envelope absent in bacteria and archaea but present in eukarya. Membrane enclosed organelles are absent in both bacteria and archaea but present in eukarya. Peptidoglycan in the cell wall found only in bacteria but absent in archaea and eukarya. RNA polymerase, one kind in bacteria, but we have several kinds in both archaea and eukarya. Initiator amino acid for protein synthesis. In case of bacteria, we have 4-methionine, but in case of archaea and eukarya, we have methionine. Introns in genes in bacteria, absent or very rare. In archaea, present in some genes, while in eukarya, present in many genes. You remember the gene splicing. Response to antibiotics, streptomycin and chloramphenicol. Bacteria growth is inhibited usually, unless we have a mutation. Growth is not inhibited in both archaea and eukarya. Histones associated with DNA, absent in bacteria but present in some species of archaea and present in eukarya. Histones in eukarya equals the amount of chromosomes, of DNA, sorry. Circular chromosome, present in both bacteria and archaea but absent in eukarya. Growth at temperature above 100 degrees centigrade, found in some species of archaea only. We refer to these organisms as thermophiles. Some archaea live in extreme environments and are called extremophiles. Extreme halophiles live in highly saline environments. Extreme thermophiles thrive in very hot environments. This figure, in this valley, there are many organisms referred to as thermophiles, many archaea. The third group of archaea are the methanogens, named for their unique form of energy metabolism. Those organisms live in swamps and marshes and produce methane gas as a waste product. They use CO2 to oxidize hydrogen to produce both energy and methane as a waste. The marsh gas found in some environments is the methane released by these organisms belonging to archaea. Other species inhabit the anaerobic guts of cattle, termites, and other herbivores, playing an essential role in the nutrition of these organisms. Methanogens are also useful to humans as decomposers in sewage treatment facilities. Methanogens are strict anaerobes and are poisoned by oxygen. The fifth section of this chapter is about the effect of prokaryotes in the biosphere. Prokaryotes are so important that if they were to disappear, the prospects for any other life surviving on Earth would be dim. Although prokaryotes are small, their large number and metabolic abilities enable them to play key roles in ecosystems by decomposing wastes, recycling chemicals, and affecting the concentration of nutrients available to other organisms. The existence of some ecosystems depend on prokaryotes. For example, the ecological communities of hydrothermal vents depend on chemoautotrophic bacteria for energy. Chemical recycling. Prokaryotes play a major role in the recycling of chemical elements between the living and non-living components of the environment. Some chemoautotrophs, prokaryotes, function as decomposers, saprobes, breaking down the dead organisms and waste products. Prokaryotes can convert some molecules to form that can be taken up by other organisms. We see before how bacteria affect the uptake of nitrogen by plants. Another example, prokaryotes can increase the availability of nutrients required for plant growth. Prokaryotes can also immobilize or decrease the availability of nutrients by using them in their own cells. Looking at this figure, which shows the impact of bacteria on soil nutrient availability, in this case potassium. No bacteria is added here. When no bacteria is added, the uptake of potassium is about 0.5 milligram. Add strain 1 to sterile soil, increases the uptake of potassium to about 0.7 milligram. Add strain 2 to sterile soil, increases the uptake of potassium to about 0.6 milligram. While adding strain 3 to sterile soil increases the uptake of potassium to more than 0.9 milligram, that's to say more than 55% increase in potassium uptake. Ecological interactions. Symbiosis is an ecological relationship in which two species live in close contact. The larger one, the host, and the smaller one is the symbiont. Prokaryotes often form symbiotic relationships with larger organisms. In mutualism, both symbiotic organisms benefit. For example, bacteria, headlights, the glowing oval below the eye of the flashlight fish is an organ harboring bioluminescent bacteria. The fish uses the light to attract prey and to signal potential mates. The bacteria receive nutrients from the fish. In commensalism, one organism benefits while neither harming nor helping the other in any significant way. For example, more than 150 bacterial species live on the outer surface of your body. Some of these are commensalists. You provide them with the oils that exude from your pores and a place to live, while they neither harm you nor benefit you. Finally, some prokaryotes engage in parasitism. In parasitism, an organism called a parasite harms but does not kill its host. Parasites that cause diseases are called pathogens, many of which are prokaryotes. Concept 6. Prokaryotes have both beneficial and harmful impacts on humans. Some prokaryotes are human pathogens, but many others have positive interactions with the humans. Mutualistic bacteria is an example of this good effect on a human. Human intestines are home to about 500 to 1,000 species of bacteria. Many of these are mutualists and break down food that is undigested by our intestine. Plus, others produce some vitamins, especially B vitamins. Now, let's see the pathogenic bacteria. Bacteria cause about half of all human diseases. Some bacterial diseases are transmitted by other species. For example, Lyme disease is caused by a bacterium and carried by ticks. Lyme disease can result in arthritis, heart disease, nervous disorder, and death if untreated. Lyme disease is shown here. Ticks spread the disease by transmitting the spirochete bacteria. A rash may develop at the site of the tick's bite. The rash may be large and ring-shaped, as shown, or much less distinctive. Pathogenic prokaryotes typically cause disease by releasing toxins, exotoxins or endotoxins. Many pathogenic bacteria are potential weapons of bioterrorism. For example, Bacillus anthracis endospores causes anthrax, جمرة الخبيثة. Another example, Clostridium botulinum toxin caused food poisoning. Prevent the release of acetylcholine from the presynaptic neurons. Another example, Yersinia pestis causing plague, ta'un. Exotoxins are proteins secreted by certain bacteria and cause disease even if the prokaryotes that produce them are not present. For example, Vibrio cholera cause cholera by secreting exotoxins that affect intestinal cell membrane permeability to ions and lead to massive diarrhea. In another example, the potentially fatal disease, botulism, is caused by botulinum toxins, an exotoxin secreted by the gram-positive bacterium Clostridium botulinum, as it ferments various foods, including improperly canned meats, seafood, and vegetables. Another example, Clostridium difficile, produces exotoxin that cause severe diarrhea, resulting in more than 12,000 deaths per year in the United States alone. Endotoxins are lipopolysaccharide components of the outer membrane of gram-negative bacteria, released only when the bacteria die and their cell walls break down. Endotoxin-producing bacteria include species in the genus Salmonella, such as Salmonella typhi, which causes typhoid fever. Food poisoning caused by other Salmonella species that can be found in poultry, دواجن, طيور, and some fruits and vegetables. Horizontal gene transfer can spread genes associated with virulence. For example, pathogenic strains of Escherichia coli contain genes that were acquired through transduction, phage-mediated horizontal gene transfer. Now we're going to talk about prokaryotes or their role in research and technology. Many foods and beverages are produced with the help of bacterial microbial fermentation. Bacteria are used in production of yogurt, cheese, pickles, soy sauce, fermented meats such as salami, vinegar, and much more. It is also used in the production of beer and wines. Bacteria is used in the production of citric acid, a compound added to candy and to many soft drinks. What are the roles of prokaryotes in research and technology? We're going to mention some of these roles. Experiments using prokaryotes have led to important advances in DNA technology. For example, Escherichia coli is used in gene cloning. Bacteria have been genetically engineered to produce certain vaccines, vitamins, hormones like insulin, and so on. For example, the prokaryotic CRISPR-Cas system can alter genes in other organisms. See next figure. CRISPR opening new avenues of research for treating HIV infection, the virus that causes AIDS. See the control cells here. Untreated cells were susceptible to infection by HIV, while cells shown here, these cells were treated with CRISPR-Cas9 system that remove HIV provirus that had become incorporated into the DNA of human cells. Bacteria can be used to make natural plastics, as shown in the figure. Bacteria synthesizing and storing compound called PHA, a component of durable biodegradable plastics. Bacteria are also being engineered to produce ethanol from agricultural and municipal waste biomass, switchgrass, and corn. Prokaryotes can also be used in bioremediation, the use of organisms to remove pollutants from the environment. This figure shows spraying of fertilizers that stimulates the growth of native bacteria that metabolize oil, increasing the speed of the breakdown process up to 5-fold, therefore removing the oil spills. Another example on bioremediation, prokaryotes decomposes organic matter in sewage, majari, converting it to fertilizers or to receive clean water from sewage treatment. Precipitating radioactive materials such as uranium out of the groundwater, another example. Also, prokaryotes can be used to inactivate radioactive materials. They are also used in mining industries such as gold mining. End of the chapter 27. I hope that these two videos, part 1 and part 2 of this chapter 27, were helpful. Thank you for listening and good luck.